In this short talk, I'll talk about the microbial dysbiosis and its relationship to epithelial barrier theory. When we mention epithelial barrier theory, we have always three major pillars. The first one is epithelial barrier damage and epithelitis starting after the barrier damage. The second pillar is microbial dysbiosis and loss of biodiversity, loss of commensal bacteria, and colonization of the opportunistic pathogens in the epithelial barrier damaged area. And the third pillar is immune system activation in response to this. And all of these events are fully interrelated and they are overlapping. Here we have the epithelial barriers, and normally microbiome floats above the epithelial barriers, and the barriers function is mainly to avoid that these microbes do not go deeper in the tissues. So this is a very historical, important interaction of this microbiome with our epithelial cells. And this is a very homeostatic interaction of several 100 million years. The first prokaryotic cells were developed around 2 billion years, and the first eukaryotic cells were developed about 1 billion years ago. 1 billion years ago, when the eukaryotic cells developed, they made an agreement with the prokaryotic cells to live together. So prokaryotic cells were living on the eukaryotic cells in the early organisms, and they were providing nutrients, vitamins, metabolites, helping the digestion, and educating the immune system. Cell differentiation was one of the main functions that they were helping, and maturation of the tissues. So the mammalians developed around 300 million years ago, and all of these functions like digestion, immune education, cell differentiation, tissue maturation are also continuing currently in humans. And so Homo sapiens benefits a lot from these prokaryotic microorganisms in our body. And we are carrying about 1 kilo of healthy microbes in our body. So this interaction was very well established. Unfortunately, nowadays, this interaction is broken, and its relationship to epithelial barrier theory and microbial dysbiosis is hampered by using toxic substances that are mentioned in other lectures. And our gut has normally 4,000 species, and the number of these species are decreased to around 2,000 when there's epithelial barrier damage and any disease development related to it. On the skin, we have around 800 species of healthy bacteria, and it's also decreased to around 300. That's what we have seen and published in atopic dermatitis. So this interaction is very important for health, and epithelial barrier theory explains that this microbial dysbiosis and loss of biodiversity, colonization of opportunistic pathogens are taking place when the commensal bacteria are lost and the epithelial cells do not benefit from this interaction anymore. Thank you for your attention for this small lecture. I thank very much to our co-workers.